I'm Alex Harrison, a third year biochemistry student, and I'll be discussing my honours project. My project has been working on trying to optimise the production of a protein in bacterial cells called ULK4. ULK4 is classified as a pseudokinase, a group of proteins that are related to the kinases, which are key cell signalling modulators. Although pseudokinases are similar to kinases, it was first thought they had no role in cell signalling pathways due to subtle but important differences. However, there has been substantial evidence of late to suggest that pseudokinases may be key in the modulation of cell signalling. This has led to lots of recent work into pseudokinases, with there being reason to believe they may play a role in cancer, schizophrenia, and other diseases. ULK4, a pseudokinase, may therefore be of scientific interest, which is why optimisation of its expression is needed. The first parts of my experimental work aimed to find out the optimal conditions for growth of the bacteria and production of the protein ULK4. 5 different conditions of different temperature, incubation time, and IPTG concentration were screened at a small scale using a strain of bacteria called P. coli and can be seen on the on-screen table. IPTG is a reagent used to trigger or induce the production of a specific protein by a bacteria. It was found that the highest yield of ULK4 protein production from the bacteria occurred when they were incubated overnight at 18 degrees with 0.5 millimolar IPTG. However, when these conditions were scaled up from the small-scale screening condition to a larger scale, yield and purity of ULK4 was poor. So different varieties or cell lines of the same E. coli bacteria were screened to see if an alternative to the P. variant would produce ULK4 in a more efficient manner. From this new small-scale screening study of different bacterial variants, it was concluded that the SL-BL21 variant of E. coli produced ULK4 with a better yield and purity than the previously used P. E. coli bacteria. Upon scale-up of the newly found optimal conditions in the cell line, there was a much improved purity and yield of ULK4, enough to allow confirmation that ULK4 had actually been produced through a technique where proteins can be recognised specifically called Western blotting. Through this, it could be concluded that the pseudokinase ULK4 had actually been produced. I would consider my research project to be a personal success, as I have acquired many new skills, enjoyed working in a research environment, and feel that my work on ULK4 can be carried on and can be of use. Given more time, more investigation can be done into whether this protein does play a role in cell signalling and maybe cancer, like other pseudokinases, now that the protocol for its efficient production has been established. Thank you for watching my podcast. I hope you've enjoyed it. Goodbye.